Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today the video is related to microbiology, and the title of my today's lecture is Triple Sugar Iron Test. And what is basically the mechanism of this test? Basically, triple sugar iron test is a biochemical test that is used for the identification of different microorganisms, and it is used to differentiate in between different microorganisms. Okay, then the microorganisms that are related to this test and that are that are examined through these tests is basically related to the Enterobacteriaceae family, that include E. coli, Shigella, Pseudomonas, Salmonella, Klebsiella, and Proteus. These such type of microorganism can be differentiated from each other through this test. Okay, so in this video I will discuss the composition of the TSI. I will discuss the different steps that is involved in the preparation of this media, and at the last when we get the results, so the main part of the video is that when we get different results, so how you will identify and differentiate in between those results, and how you will manage to understand those results. Okay, because there are two three type of results in present in that test tubes. Okay, so the main identification and the examination of their those results are very much important. Okay, so let's start with the video, and I want to tell you that please watch this video till end so that you are cleared about the TSI test. Okay, so let's start with the video. First of all, you should know about that what is basically TSI. It is basically known as triple sugar iron agar media. It consists of dextrose, lactose, and sucrose. That's why it is known as triple sugar. And why it is called iron? So iron is basically ferrous sulfate is basically present in it. It is performing a specific function. I will tell you that what functions are performed by these sugar and iron in the later video. And also it consists of agar in order to solidify this media. So that is why this is known as triple sugar iron agar. And why it is used? It is used to differentiate in between the gram-negative enteric bacteria. Okay, so these bacteria produce fermentation, perform fermentation of dextrose, lactose, and sucrose, and produce different colors. Okay, so and we then examine those colors, and then we write the results. Okay, so that's why it is known as triple sugar iron agar. What is the principle of it? So basically, again, I'm telling you that it is used to identify Enterobacteriaceae, the species of Enterobacteriaceae, and it is used for the identification of gram-negative bacilli. Okay, so if I come to the composition of it, so triple sugar iron media consists of beef extract, peptone, and egg peptones, and yeast extract. These three components are basically used to provide nutrients and provides amino acids and different other nutrients to the microorganism. Okay, so beef extract, peptones, and yeast extract are used as a nutrients. Lactose, sucrose, and dextrose monohydrate, or it is simply known as D-glucose, is used for the fermentation process, and ferrous sulfate is used. I will tell you the main function of the ferrous sulfate, and sodium chloride and sodium thiosulfate and is also used. And at the last, phenol red is also used. Phenol red is basically used as an indicator. The actual color of the TSI media is red, and when there is shift in the pH, so different colors are produced. So that's why we use phenol red. So when other colors are produced, so then when we examine those colors. Okay, so phenol red is used as an indicator, and at last agar is used in order to maintain the solidification of this media. Okay, so keep these terms in mind. If again I tell you, so as I have tell you that peptone, yeast extract, and beef extract provide nitrogenous compound, sulfur, and trace element and vitamins to the microorganism in order to grow them. and sodium chloride maintain osmotic equilibrium, and lactose, sucrose, and dextrose is used as a fermentative, used for fermentation. Then sodium thiosulphate and ferric or ferrous ions make H2S, and basically the production of H2S is also an indicator. Okay, and this is produced by specific bacteria. And as I have told you that phenol red is used as a pH indicator, and so sodium thiosulfate is also used to minimize the toxicity of the microorganism. So these all were the composition of it. If we come to the preparation of it, so the preparation is very much easy, and this preparation is mostly known to the most of the microbiology student. You have to take a specific amount of media and dissolve it in a specific amount of water. If you want to take 1 liter of water, so you have to dissolve 64.42 grams of the TSI media. And if you want to decrease the level of the water, so then there is a specific formula for that. You have to just divide and multiply, and you have to get the actual amount. And I'm pretty sure that you know about it. And after that, and now this is the process. You have to then autoclave it in order to destroy all the microorganisms that are present in the media and on the beaker and other. And after that, you have to pour that media into test tubes. And keep this thing in mind that you should also autoclave the test tubes. Okay, the glass tubes. Basically the chemical biochemical test is performed inside the test tubes, so test tube should also be autoclaved. Then you have to pour the media into the test tube. It's on your choice that how much test tube you want to take it. It rely on your experiment. Okay, for example, then we have poured our media into the test tube. Then you have to put the test tubes a bit curved. Okay, so that's no so that's why so that slants should be produced. These are known as slants. Okay, so if you pour the media and after that you place the test tube like in a curved way, so these slants are produced, and these slants are very much important for the biochemical tests of the TSI. So that's why when it solidifies, so it produces slants. So these are known as slants. Okay, after that, if I want to show you the actual picture of it, so this is the tube in which the TSI media is present. And again, I'm telling you that the actual color of the TSI media is red. Okay, because phenol red is present. After that, you should take you should take a specific colony, unknown colony, on a wire loop, and then you have to first this is the actual and just look here. You have to place you have to dip the inoculating loop into the media, and keep sure that you should not touch the end of the test tube. Then after that, you have to just gently remove the inoculating loop, and when you reaches the slant, the upper surface, so then you have to make a small curving like position. Okay, you have to just curve, or you have to just streak over the slant. And after that, then you have to remove the inoculating loop. One thing, keep this thing in mind that this portion is known as butt. I'm just writing B. Okay, so this portion is known as butt and this portion is known as slant. Okay, keep these two terms in your mind. So after this, we will place those tubes in the incubator, and on the next day or after 2 days, you can see the result. So 4 types of result we can get 4 types of result. So the main thing is that we should understand about the result, that what does these means. If there is yellow color, if there is multi-color, if there is black color, or if there is no color, so what does this mean? So basically the video is about this. So understanding the results. So first of all, if we get oh wait a minute, one thing you should know, that if the media is totally red, so this means that the media is alkaline. Okay, and if we it convert into, if there is fermentation and acid is produced, so it will completely turn into yellow color. Okay, it will turn into yellow color. So if there is yellow color in the media, so you can easily tell that there is acid production, because the acid lowers the pH, and due to the lowering of the pH, yellow color is produced. The yellow color, the red color is transformed or changed into yellow color. So keep this one principle in your mind, that red color means that the media is alkaline, and yellow color means that the media is acidic. Okay, and mostly the acid production is and the acid production is here in the butt region, because there is lack of oxygen, so the microorganism mostly preferred fermentation. And here respiration takes place, because here is sufficient amount of oxygen, so that's why mostly respiration takes place here. So in case of here, there will be more acid production with respect to the slant position. Okay, so keep this other plants principle in your mind. Now if we get this such type of result, okay, so what does this means? So this means that we are having dextrose, lactose, and sucrose in our media, and all of the three sugars are fermented by the microorganism. So if all the dextrose and lactose and sucrose is fermented, so large amount of acid is produced, and just because of that acid, the complete media is converted into yellow color. So this means that all the microorganisms that are present in this media has ferment glucose, sucrose, and this dextrose. So that's why acid is produced. And here is, just look here to this portion, it is just here is the production of CO2. Okay, because in the fermentation, carbon dioxide can also be produced. So this CO2 has moved the media to upper direction. So here we can easily tell that here is CO2. Okay, here is CO2, and just because of this CO2, it has moved the media to the upper position. Okay, it has moved the media to the upper position. So when you see such type of test tube, so you can easily tell that the yellow color means that all the three sugar compounds are converted into are fermented, and and acids are produced. And this portion, when you see this portion, so you can easily tell that carbon dioxide is also produced. Okay, so hope you have understand this result. If you get this such type of result, so it is a bit complicated one. Okay, just to open your ears, because this result is a bit complicated. That if we are having a bacteria that can only ferment dextrose, okay, if it ferments only dextrose, so such type of condition can occur. This is the normal case. Okay, this is the normal one. And if this is the normal one, and if this such type of result come, so what does this mean? This means that these bacteria, the bacteria that is present in this test tube, can only ferment dextrose. Okay, that's why this such type of result appears. What does this mean? That in the TSI we are having dextrose, that is basically zero in the concentration of 0.1%, and we are having sucrose, that is 1%, and lactose 1%. But here the dextrose amount is a bit lower, and it is 0.1%. Okay, and another thing, keep this in mind, that when the bacteria is dividing, so they are performing decarboxylation reaction, means that they are continuously producing CO2 in the Krebs cycle and in the electron transport chain. Basically in the Krebs cycle, carbon dioxide is produced. Okay, so they are also producing carbon dioxide. There are also other chemical reaction just like this reaction, that the amino acids are converted into amines and CO2 by the decarboxylation reaction. So just look here, I'm showing you this compound. This is basically known as amine, and when there is amine is basically alkaline. Okay, so continuously when the bacteria is dividing, so that bacteria is continuously producing amine. And also the bacteria that only ferment, due to the fermentation it is producing acid. So at the same time acid is also producing, okay, and bases the amine are also producing. But one thing is that acid is a bit in higher concentration, okay. For example, if the acid is 50%, okay, if the acid is for 50%, so the amine that are producing is about 30%, okay. But when there is alkali and there is acid, so they are both are neutralizing their effect, okay. So this is just because this such type of result is present. Just look here. Here if you look here to the yellow color, okay, so it is not that much bright because here is acid, but it is in lower amount because that acid is neutralized by the amine. And just because of it that the dextrose is in lesser amount, so dextrose is being fermented by that bacteria, but it is in lower amount. And that bacteria cannot ferment sucrose and lactose. So the sucrose and lactose remains the same, and that bacteria is continuously fermenting dextrose and producing acid. But that acid is not in very high amount with respect to when the sucrose and lactose was fermented. But continuously the bacteria is also dividing and it is also producing amine. So these two compounds are neutralizing each other, although the concentration of acid is a bit higher. So that's why a pale yellow color is here, okay. A pale yellow color is here, not bright color, okay. If it was bright, so it would mean just like the first case, that it would mean that it has completely ferment all the sugars. But here is a pale yellow color, and this is just because this amine is continuously neutralizing the acid. And here and now here that amine has not completely but partially neutralized the acid. So this such type of result is present, and this means that when the bacteria is only fermenting dextrose, so just because of it this such type of thing happens. And the yellow color will be pale. So here if I tell you that 50% is acid and 30% is uh, base, okay, 30% is base. So they both are neutralizing each other, although the concentration of acid is 20% higher. So that's why it is producing pale yellow color, and this portion is now much paler. Means that here is yellow color, but it is not that much in high amount. So this is just because the bacteria is only fermenting the dextrose, and it is not fermenting the sucrose and the lactose. So that's why this is the main reason. And mainly due to this result, the students are facing different difficulties and confusion. So hope you have understand it. Again, now this is the text form, that growing bacteria also form alkaline products, that is known through oxidative decarboxylation of peptones. Peptones are the amino acids, and oxidative decarboxylation means when the carbon dioxide is produced, okay. So it is known as oxidative decarboxylation, and the oxygen is also involved, okay. So the appearance of an alkaline red slant and an acid yellow bud after incubation indicates that the organism is a dextrose fermenter, but it is unable to ferment lactose and or sucrose, okay. So this is a text form and I have explained it. And if we get this such type of result, so what does this means? That this black color, this means that the thiosulphate is reduced to hydrogen sulfide, okay. And this hydrogen sulfide reacts with the H2S, and this FeS, black precipitate of ferrous sulfide, is produced, okay. And this the color of the ferrous sulfide is black in color. So that's why this color appear. So when this color appears, so you can easily indicate that this bacteria has produced ferrous sulphide. Okay, this bacteria has produced ferrous sulphide. It has not ferment glucose or that dextrose and sucrose. It has only produced the thiosulphate is reduced to hydrogen sulfide, and after that by several species of bacteria, and hydrogen sulfide combined with ferric ions, okay, that is present in that media, and it produced an insoluble compound that is known as ferrous sulfide, and that ferrous sulfide is black in color. So that's why, and the reduction of thiosulfate proceed only in an acid environment, and blackening usually occurs in the butt of the tube, and this is basically the acid. Okay, the H2SO4, this is the acid. But what happens that it produces this black precipitate of ferrous sulfide. So if someone asks you that why this black colors appear, so you should tell them that it is just because of the production of H2S, and this H2S combines with the ferric ions and ferric salts, and ferrous sulfide is produced. Okay, so keep this thing in mind. If we summarize our entire chemical reactions, okay, so if we are having an alkaline slant and acid butt, okay, if the slant is red in color and the butt is yellow in color, so this means that only glucose is fermented. Okay, only glucose is fermented. And if we are having an acid slant, okay, if it is yellow in color and also an acid butt, so this means that dextrose and sucrose, or dextrose and lactose, or even dextrose, lactose and sucrose is fermented. Okay, if we are having bubbles, so this means that gas is produced, and then that gas is carbon dioxide. Okay, and if we get a black precipitate, so this means that hydrogen sulfide is produced. Okay, so this was a summarized form of the chemical reaction that I have told you. Now if we are having four tubes, okay, so tube A contains of alkali genes species. So this it has not performed any such type of fermentation, so the entire test tube is red in color. Okay, so there is no fermentation, no gas production, and no H2S production. And if we look here to the B tube, and it is E. coli. So E. coli can ferment dextrose, sucrose, and lactose. So completely all the test tube is in a yellow color. And just look here, okay, here is a CO2 production. Okay, this is the CO2. If we look to the test tube number C, okay, this C, so it is Salmonella typhimurium. So it has produced H2S. Okay, it has produced H2S, and it has also ferment the glucose and the sucrose and the the dextrose. And so this we can say that it is Salmonella typhimurium. And if we look to the tube D, so tube D has only ferment glucose, or simply dextrose. Okay, so this was the case that it has only ferment the dextrose. This is the result book. Okay, if you can compare, you can compare your test, your result with these such type of thing, this type of the, if you just look here to the TSI portion. Okay, this is the TSI portion. So if we are having slant, and this negative sign mean that no production, and positive sign mean that production. Okay, so if you are having, just look here, slant, so when there is color on the slant, so you can say that it is positive. If there is no color on production, so we can say that it is negative. If we check on the color of the bud, so if there is color, so positive. If there is no color, so negative. If we look here to the gas production, so if there is gas production, so it means that plus. If there is no production, so it means that there is it is negative. If there is hydrogen sulfide production, so it means negative. And if there is no if there is hydrogen sulfide production, so it means that positive. And if there is no hydrogen sulfide production, so it means negative. Okay, and these all are the species that are that are giving these such type of results. Okay, and now here is if you get the results, okay, so you can enter your bud color and the slant color and the carbohydrate fermented and the blackening and the H2S production. Okay, so you can fill these table and you this table can be given to you by your examiner. Okay, so this was all about the how the triple sugar iron test. Okay, and I have explained each and everything. Still, if you have any further confusion in it, so you can text me, sorry, you can comment me in the comment box. I will answer you there. If you have understand this video, so like this video, share it with your friends, and subscribe to my channel for more interesting videos related to science. Thank you very much. God bless you.